Hey everybody, it's Miss Chotzi. I hope you're doing well. I wish I could see you, but I can't. Hopefully you've been keeping up with the work. Thank you to everybody who has been keeping up with the work. I don't mean to pile it on. We just have a few things left to do this year, and then we'll pretty much just be doing different kinds of reviewing. I did get details on the updated AP Bio exam, so I'll have another little video for you on that one coming up. We're gonna do it. We're gonna be great. You're gonna be great, and I hope everything is going great in your life. So, thank you again for keeping up. Okay, so we have one last little assignment here in photosynthesis to accomplish. We would have been doing a lab together, but that's not happening. So instead, I'm going to have you watch a little video that another teacher put together of the actual lab, like the procedures and how it works. I'm going to have you kind of analyze a little bit about what they're doing, and then our version of it is we are going to do a little simulated lab where you're going to create some experimental setup and then create some data and analyze what you think that data is going to mean. One of the FRQs on the AP Bio exam this year is going to be focused a lot on experimental design from what I have heard. So I want to make sure that we do an example of that. So to begin, make sure you watch this little video here about this particular photosynthesis experiment. It's only about 7 minutes long, so it shouldn't take up your whole life. While you are watching the experiment, I put together some questions for you about what's going on in the experiment, and then just some review questions about design. So you're just reviewing, what do plants need and what do plants make. You're going to look at some of the equipment that they intend to use in this experiment. One of them is a syringe, so you'll understand hopefully the purpose of that syringe. But what I really want you to look at and focus on are, can you identify the independent and dependent variables in this experiment? So the independent variables are, what is the one thing that the teacher is manipulating and controlling and making different between different experimental groups? And then the dependent variable, of course, is what is the data that it's a guy, that he is collecting during the experiment? What is changing over time? I also want you to be able to identify a constant in the experiment. A constant is different from a control. A control is the group under normal conditions. I could argue that this experiment doesn't really have a control group, but it definitely has a constant, and many of them. So these are just things in the environment that you have to make sure stay the same for all of your experimental groups. Because if there's something that is not constant in between those groups, we would identify that as our independent variable. You can only have one, so you can't have more than one of those. Also, a few questions about what's going on. The essence of the experiment is that you've got a plant, you've got spinach leaves that have been punched into little tiny paper discs, like with a hole puncher, except not made of paper, but made of spinach. And over time, they're going to sink and then they're going to float, and you're going to figure out why that happens. But that is an indicator of the rate of photosynthesis. Okay, so there's a few more questions to answer about what's going on in the experiment. Okay, then I want you to take a look. I took the gentleman's experiment and made up some fictional data about what I think was going to happen in that particular experiment. So I want you to take a look at this data table. The experiment runs for a cumulative time of 15 minutes. And then what you see in the two columns are the number of leaf discs that were floating at that given time. So it is also a cumulative number. I believe in his experiment, he only ever went up to 20 discs per group. So that's why my number ends at 17. And you can see that in this experiment, he was measuring what happens to the discs under light and then what happens in the other experimental groups that are kept in the dark. And what I would like you to do is write a statement. It just really only has to be, a sentence or two at the most, about what the data is telling you. So look, what's the trend in the data? What's the relationship between light and dark? Why are the light numbers doing this and why are the dark numbers doing that? And you can use your knowledge of, what you know about photosynthesis. Okay, so number 3 is just about drawing a conclusion based on the data. Definitely one of the skills that's going to be on the AP exam, I think. Okay, next, this would be where we're going to begin designing your own experiment, but unfortunately we can't do it, but we can set it up. I want you to brainstorm a list of 3 other things that you could test in the parameters of this experiment. So using the same setup, mostly. I will give you a hint if you need a little inspiration. Think about what plants need, think about what plants make, but particularly what they need. So can you manipulate any of those three things? In his experiment, he did light versus no light. Is there anything else you can do with light? Can you do anything with carbon dioxide? Can you do anything with water? Okay, so just make yourself a list. Here's one example. If I could, since I know that plants need CO2 to do photosynthesis, if I manipulate or change the concentration or the amount of CO2, can I influence the speed or the rate of photosynthesis? So you're going to make your little list here. And then I want you to, in number 5, pick one of the things on the list, whatever one you think you know the most about or you feel the most comfortable with. Just choose one. And that's going to be the source then of your experiment. So your experimental question is, how does this variable change the rate of photosynthesis? What I want you to do is make a claim about how you think that variable is going to affect the rate of photosynthesis. And remember, rate means the speed at which it happens. So essentially it would be like how fast do plants grow? Now, I would recommend using the if-then format for your claim, just like a hypothesis. So for example, in the previous sample I gave you up here, if I'm manipulating the concentration of CO2, a claim might be something like, if I increase the amount of CO2, like if I double the amount of carbon dioxide, then the rate of photosynthesis will, and then I would say increase, decrease, or stay the same. I'm going to go with increase on this one. So then I will write that claim out. Then the fun part is you're going to actually make some BS data. Okay, so you're going to just totally BS some data here that is going to support your actual claim. Okay, so it says create some fictional data that supports your claim and complete the following data table. Make up the numbers to show how your variable should affect photosynthesis. So if I'm saying that if I double the concentration, I should increase the rate of photosynthesis, I'm going to be putting numbers over time here that are going to show that happening. Okay, so even if it's not right, I just want you to, using the part of your brain that can make up data to support something is a pretty sophisticated skill set. So it'll help you with analyzing charts and graphs. So here you're just making up some data. Make sure that on these lines here, you tell me what your experimental groups are, like this one's getting this and this one's getting that. And then show me your numbers. And you don't have to go past 15-minute mark. Remember that you're not using more than 20 discs per column. Okay, so just keep it in line there. Okay, so you make up the data. Then the final step is to do what we did earlier, where you have a data table. Now you have to draw some conclusions based on it. So I want you to write a conclusive statement, like a conclusion. It only really needs to be a sentence or so. Just giving me the overall results. So over time, this is what happened here, and this is what happened there. The next part is to begin to explain why. So explain why your data makes sense. Why is the rate of photosynthesis changing? So now you use your knowledge about photosynthesis and your variables and explain to me, the why. Why does increasing the amount of carbon dioxide, increase the rate of photosynthesis? What's going on there? What's going on at the cellular level? What's going on in the chloroplast? And then lastly here, this is a big skill I'm pretty sure is going to be addressed on the AP exam. They want you to use data that's provided to support your claim. So in freshman year, we did claim evidence reasoning. It's basically the short version of the CER. You're going to pull some data off of your fictional data table, like, oh yeah, these 2 data points really, tell me a lot of things. And then you're going to reference them in the answer. So you might say like, at minute 1, this is what I saw, but then at minute 10, this is what I saw. And that separation or those differences in numbers shows me, X, Y, and Z, that this is what's happening in photosynthesis and this is why. This would probably be, of the 3 things on this page, this one would probably be the longest. And by long, I mean maybe 2 to 3 sentences. Like, please keep it short. The idea here is you shouldn't need a lot of words to say this stuff. You're supposed to be very concise in your meaning. If you're saying too much, if you're writing too much, you're probably being redundant and maybe rambling a bit. So try to keep your statements as clear as possible. Okay, so that's our last little assignment on photosynthesis. We have a few more things next week with respiration, but it's a lot shorter. So we'll talk soon. And then after that, we'll just start reviewing for the exam. Okay, let me know if you have any questions on this one. Take care.